Pleasure to have Dennis Wang. So Dennis has a joint appointment at Imperial and starts in Singapore. So he's a professor at the National Heart and Lung Institute in Imperial, and he's also a senior principal scientist at start Singapore. So thank you.
Thank you. Okay, welcome back. So I'm based, like Mauricio said, I'm based at two translational medical institutes. And since it's kind of last talk of the day, I decided to kind of have a slightly different presentation, hopefully a little bit lighter, not as much energy intensive. And I'm going to mainly go a bit high level in terms of giving an overview of kind of applications of ML. And I guess the learning objective here is I just want you to go, hopefully inspire you or show you some interest in some different applications of ML. And I, looking at the posters and speaking to some of you yesterday at the dinner, realized people are, working in applying ML to various different domains. So hopefully this may inspire you to kind of apply your methods in another medical domain, or develop methods in one of those domains. Okay. So first, I want to look at kind of what is our objective here and what we're trying to maximize and minimize. But first, we want to test our kind of assumptions. So this is just a quick survey. How healthy you think the world is, not on an ecological sense, but in terms of the human population. So first question. So you can just raise your hand here. So worldwide, how many babies are born with a trained health worker? So if a baby was born, are they, is there like a healthcare professional there? Who thinks it's A? Okay. Who thinks it's B? Pretty even. Who thinks it's C? Oh, okay. Okay, great. Let's see if our assumption is right. So I think everyone's assumption is wrong. So gotta be careful if we're gonna be developing an epidemiological model on birth outcomes. So even worldwide, I think statistically, the infant mortality rate has been drastically decreasing, and this has in every part of the world, right? And this has something to do with it. I think we are just a lot better kind of medical knowledge around what determines good birth outcomes, and so that has affected population health. But you're not most of the population in the UK. Answer the same as you, so you're not an outlier. You're not an outlier, so that's good. Okay. Next question: how many of the world's 1-year-old children were vaccinated against some disease in 2019? So just before COVID, vaccination rates went up after COVID, but just before COVID, how many do you think? A, less than 20%?
Are we allowed to use what we just found out?
You can have, you can, change your prior. It doesn't matter. B, around 50%. Okay. And C, more than 80%. Okay. So it's a bit more split today. So a bit more split. The world is healthier. So most people, vaccines a great invention. Again, like childhood diseases, childhood mortality has gone down quite a bit all around the world, right? Because we have very cheap vaccines to some of the most horrendous diseases like polio, TB, and so forth. Okay. The first two questions around child, and just a little bit background. One of my institutes in Singapore, we look at child health, so that's why I selected those questions. But we're looking a little bit now at adult ages. So in low-income countries today, the low-income countries today had a life expectancy of 44 years back in 1970. What is it now? So what do you think is the life expectancy of low-income countries now. 40 years? 50 years? That's 50. 60 years? Yeah, almost. So I think people are getting a hang of this question. So yeah, 60 years. So even in low-income countries, health outcomes have gotten better. Generally, even though there is a lot of inequality in the world, on average, like the world has gotten wealthier on average, and like healthcare capacity has gotten better across the world. Okay. All right. So a little bit more on demographics. So depending on, this is important because I look into a little bit of global health, and depending on where I am, which continent I am, I have to adjust my assumptions. So in Pakistan, how many people, is anyone from Pakistan? Oh, great. So you should know this. We'll check up on that. How many people in Pakistan are considered elderly? I guess 65 and older. In the UK, you could collect your pension by then, but now you have to work a little longer. But around 5%? Or 5? Okay. Around 17%? Okay. And around 29%? Okay. Yeah, it's pretty split, this one. So it's actually 5%. It's a very young population, actually. So yeah, not many elderly people. Okay. Okay, hello. Getting a little bit more depressing now. Okay. So I know people have probably heard the news of mental health being a kind of epidemic these days in a lot of countries. So what do you think happened to the global suicide rate in the last 20 years? Decreased about 25%? Okay, or stayed about the same? Stayed about the same, yeah. Increased about 25%? Okay, yeah, so I see I influenced your decision. I biased your choice there with my little comment. So yeah, it's actually, the suicide rate has actually decreased. But, it's still I think maybe because of social media and stuff, I think maybe the news of it is maybe more impactful and stuff. It's still, it doesn't say it's not a problem, right? But I think, sometimes the statistics might be different from what we perceive. Okay, so that was the quiz, by the way. So a little bit kind of summary about kind of those statistics. So one is around aging, right? Given this ELLIS, and Germany is a European country, we can see that in 1950, right after the war, it's more of an impoverished, lower income country, right? The population is quite a bit younger, right? And then now the demographics has shifted up, right? So majority of the population is middle age or elderly, right? And then there's a large health burden there. But the other thing to notice is actually, even though a lot of people are, needing healthcare, more healthcare services, there's also proportionally less working adults, right? So there's also a social problem here of there's less people to support this healthcare system. So that might mean we need more intelligent systems to kind of make the healthcare system more efficient and effective. Okay, and then the problem is probably going to get worse in 2034. Basically, the mean age is going to be 68. Okay, so because of this kind of aging, the change in demographics, we're seeing more and more kind of diseases that are not so acute anymore, right? They're like kind of chronic long-term diseases, and people are dying from these long-term diseases in Europe, right? Before it was infectious diseases early on when vaccines weren't as popular, but now with vaccines, with the population living longer, there's more effective treatments. We're seeing people living and then eventually dying of more chronic, long-term, complex conditions. Okay. So what I study, what I've been focusing on more is kind of not so much like a single disease, but looking at people who suffer from multiple conditions. Okay? And basically the classic kind of public health question is, can we identify patterns or determinants of health outcomes over time, okay? Over a lifespan and over a population. Okay? And when you look at populations over time, we see that actually, yeah, people do have a combination of conditions, and those combinations of conditions do change over time as well. Okay? So that's, I think, just something to be aware of. Like I thought when looking at the data, that it's a lot of times, it's just actually a snapshot of that person at a particular age. They might be suffering by the condition, but there may be actually future conditions that might also influence their health outcomes. Okay. So the basic question we're trying to ask is, how do health change as we age, and can we predict this? Right? Okay. So this is actually kind of a computational question that I think like every parent has done of it. So I'm not sure if how many parents are in the room, but when you have a kid in the UK and many countries, you have this log book where you actually measure, the weight and height of your child, and you plot it on this growth chart. And this growth chart has bands in it. Okay, and depending on where your child falls, they might be at risk for future conditions. Okay, so every individual is actually doing that data collection and forecasting of their children. Okay. And so we want to kind of do it more on like a larger scale. So generally, when we're looking at kind of risk factors for multiple health conditions or potential therapeutics to treat multiple health conditions, it's kind of a data integration problem of different high-dimensional data sets across time that include like genomic data, electronic health records, and other types of phenotypes you can collect, say from like wearable devices and so forth. Okay. Just a note, like there'll be a lot of different visualizations here, so just stop me. I tend to ramble on and on, so if anything's unclear, just put up your hand and ask a question. Okay. So just generally the process of collecting this data and doing modeling with it is generally like an iterative process where we have a cohort and we interact with the clinician to collect samples from the disease or population cohort. We work with biologists to extract the samples, do some profiling of the samples, right? And there's some data processing and linkage of data from like, say, molecular data with clinical records. We do some modeling, and then we check the performance of our model and our interpretation of the features in the model, right? And get feedback. And if we're doing kind of longitudinal study, we'll do this over time, right? So the idea is to collect more and more data and improve our models. All right, so that's just a general process. And now, so there's the second part of my title was called therapeutics. Okay, so therapeutics is actually a pretty wide term. So I'm going to briefly go through kind of different types of therapeutics and potential computational problems in the development of those therapeutics. All right, first one is drug therapies. So this is a little bit signposting part with the talks tomorrow that may talk about a bit more on drug development. Drug therapies is basically a compound, right? Someone takes, ingests, and then the kind of machine learning problems is that we want to collect usually molecular data because this is a compound. We try to collect molecular data of the patients and clinical features, and we usually do two things generally, like try to stratify those patients, do some clustering to identify patients who may benefit or do not benefit by a drug, or do a classification, right, to predict the response of the drug. Okay. another type of therapy now that's like quite new and very expensive, but it has like quite good implications for rare diseases is gene and cell therapies. So this is where, the patient, basically you extract biosamples from the patient. They're usually like cells, like say white blood cells from the patient, right? And do some genetic engineering to make it attack the pathogen, the disease tissue, right? And then inject it in the patient, right? And then you're using basically the patient's own cells to treat the disease, right? But you have to do some engineering to those like immune cells, right? So the problem here is like for say cancer therapies, or they'll engineer T cells to attack tumors, right? So the problem is try to predict the peptide sequence on the tumors that could attract T cell binding, okay? And then allow the T cell or other white blood cells to attack the tumor, okay? So the problem is basically you're getting T cell receptor sequences and trying to predict what type of sequences might bind to the tumor. Okay. Oh yeah, I'll share these slides as well, so no need to take notes of the paper, so you can go read more about these studies later. So the next type of therapy is getting quite a bit of traction, is actually nutritional therapy, and nutritional supplements. So this is kind of, I guess, the promise of this is that there's like a quicker kind of development pipeline than drug therapies, because nutrition, like usually foods are already safe. People are eating them already, right? So they still go through some of the regulatory pipelines as drugs, but maybe not as rigorous because they're already considered safe. But like, people say that you are what you eat. It's not exactly true. It's more like the microbiome is what, your microbiome is like what you eat. So the main kind of data you're learning from is gut microbiome and metabolites that are circulating in the blood to see the effect of a nutritional intervention. Okay, and so this study here has looked at basically two different ways of collecting microbiome from the gut. One is actually through a device that you ingest and travels through your gastrointestinal tract and collects microbiome, right? And the more traditional way is just collecting it from the stool, right? So you can imagine stool may have a different composition of microbiome because it's kind of the end stage of your digestive tract, right? But so it might not be representative of what's actually in your, say, stomach or small intestine, right? So the cognitive problem here is basically like kind of segmentation and clustering. You're trying to identify different populations of bacteria, right, in those samples, okay? And try to associate those samples to the type of nutrition you get, right? And then there's the downstream study of trying to associate populations of bacteria with like an outcome, like say diabetes or even Alzheimer's disease. So there has been quite a few studies linking gut microbiome to various diseases of other organs. All right. The other type of intervention is, I guess it's quite common. It's basically having surgery, right? So surgery is also a therapy, right? So a therapy is anything that kind of makes you better. So I guess the advancement in therapy, surgical therapies, is that to do like less invasive surgeries, right? So one type is like laparoscopic surgeries, where they're basically just poking a hole and doing all the surgery underneath the skin, right? So this presents a problem because the surgeon cannot see what they're cutting, right? So this becomes kind of a little bit like a computer vision problem, right? So they do have cameras in these surgical tubes, and then you have to kind of predict, because all your organs are not like spaced out on a 2D plane, right? They're on top of each other. So there is some kind of modeling and prediction of where your surgical instruments are and what other tissues you might be hitting. All right, so this kind of computer vision problem, and they also use it to. They also use these approaches, say like using augmented reality to train surgeons. So there's also you can think of you want to train surgical residents without them having to operate on a patient, right? So there's development of like kind of augment like virtual headsets for trainees to operate. And then there's also a data integration problem of you have a lot of sensors on those patients while they're on the operating table. So how do you like integrate all these measures of heart rate and measures that the anesthesiologist may be taking in terms of how much drug anesthetic is given to the patient, right? So they want to predict whether, it's always kind of a fine line for anesthesiologists. You don't want to give too much drug that it causes some poor outcomes for the patient, but not give too little drug that they feel the pain, right? All right, and then and then there's digital therapies, which my team is starting to get a bit more experience on, and this is around kind of monitoring of patients outside of the clinic, right? So, and actually treating them at home or outside of the clinic or hospital, right? So this one, I'll give you a bit more details on this study, but you can basically track longitudinally from like smartwatches, smartphones, activity of patients over time, and this activity can be predictive of the patient's like cardiovascular condition, right? So if patients have some cardiovascular diseases, respiratory conditions, you could track them over time, and then you could provide some interventions. So the therapy aspect is like, for instance, if someone isn't getting their, I think most of you, how many people here own smartwatches? Or yeah. So okay. So some of you may know what I'm talking about. So if you're not getting off your feet, sometimes they might, the phone might vibrate, tell you to like get your step count up and so forth. So there has been studies looking at the effect of these kind of prompts on cardiovascular health. So in Singapore, there's a large kind of nationwide study of like 2 million people, whereby there's actually a government app where you can opt into sharing your data on, like, say, your step count and so forth. And then the intervention is that the government will give you some prizes if you reach a certain step count. Okay, so there's like, for Singapore, there's like it works. The healthcare system works off of like a government subsidy. So if you go to a hospital for a condition, they might give you more subsidy or less subsidy based on your inputs here in terms of your physical activity, right? So if you're working out trying to reduce your risk factors, they might give you more subsidy, right? Because you're generally a healthy person and you're just kind of unlucky that you arrived at the hospital, right? But they're currently like doing kind of a large scale trial to see if this kind of it's kind of like a behavioral intervention would actually change health outcomes. Okay, so next I'm gonna talk. Okay, I'll pause here. Any questions regarding the different types of therapies before I go into one specific type of therapy, maybe drug development? No, everything's clear? Okay, good. All right. So the drug development process, I'm going to go through the drug development process because it's probably the most kind of mature therapeutic development process. And, there's a lot of regulation around it. And some of the other processes may be slightly different, right? But there are some similarities to this process. Okay. So I think previous speakers have alluded to that this is kind of a very costly process, right? And it's really a large funnel where you're starting off with product development, trying to develop compounds, and then you go into preclinical testing, and then very expensive clinical trials, and you only get a few products that kind of make it to regulatory approval. Okay. All right, so this funnel previously has been like more closed, right? Like pharma companies would have a portfolio of drugs, and then they push it through this pipeline. If you make it onto market, we're seeing now that it's a little bit more permeable, this funnel. So there's, I think, more collaborations. So there's products that they've developed outside of pharma, like at universities, that also get licensed into pharma and make it out. And some products that may not succeed, but it could be licensed to other entities, maybe repurposed for other conditions and so forth. Okay. So there's quite a few like kind of initiatives, open innovation initiatives set up by various pharma, and basically you can apply for these collaborations. Some come with data, reagents, even some funding, right? But most pharma now are quite open to collaboration. I know there's a few pharma people here, so he could give you more information about this. All right. And so, yeah, a few years ago, some of the big pharma, like AstraZeneca, looked at their portfolio in terms of what drugs make it onto the market, and reviewing kind of what clinical and preclinical evidence were predictors of success of those drugs. And some of those predictors are like there's confidence in how they select the patients, or there's kind of linkage between the target of the drug and maybe diseases of the disease of the patient, right? And then there's kind of good efficacy at the preclinical stages and so forth. So basically, it's talking about identifying the right patient to the right target and the right dose. Okay. So first, in terms of identifying the right target, a lot of people start off with genomics, right? So there's a lot of large population cohorts now with whole genomic data. So you can easily do association tests between genetic variants or genes with specific diseases or health conditions, right? And earlier there's been a few talk about causation, and for epidemiologists, they often use a technique to infer causation called Mendelian randomization. Basically, it's using a statistical instrument or proxy variable, basically the variant, to measure the causality of a target on a disease. Okay, so for those who don't know about MR, the basic idea is that the target, right, you're not sure if the target may be causal for the disease or the health condition, because there's a lot of confounding variables, right? Even environmental exposures that happen throughout the person's life that can influence this association. But the genetics, your genetic traits are decided at birth, right? So we know that there's nothing in the lifetime that could influence that, right? So if these assumptions hold, there's no association, right, between the variant and the confounder or with the outcome, and sometimes there are like other genetic variants that are also associated with the target. If these assumptions hold, then you can use MR, but oftentimes with a lot of complex chronic health conditions, this does not hold, right? Like diabetes or dementia and so forth, because there is a lot of other environmental factors that may influence the confounder and this relationship between the target and the outcome. And also there's what's known as like pleiotropic effects, that the variant may also influence a lot of other processes that eventually also influence this confounder. So this assumption does not hold either. Okay. But that's one method. I think there's kind of room for development of more causal inference methods to infer causality of a drug target to outcome. Okay. So other approaches is that has been alluded to by previous speakers, is kind of building more like mechanistic model. So like building kind of like a causal network of potential targets. So this is an example of essentially a network of proteins linked to each other in a acute myeloid leukemia cell. That this model basically predicts two outcomes: either proliferation or apoptosis or death of the cell. Okay, and so the idea, like building these cellular models, is that you could then like perturb some of the nodes in this network model and then see how it affects the outcome, right? So through kind of in silico perturbation experiments, you can potentially see if your predictions of outcome are right or wrong. And then this has been further developed into incorporating more machine learning. So instead of a kind of a mechanistic ODE model, you have an algorithm that helps optimize the parameters of your ODE model. Okay, so you can learn the difference between your predicted and observed outcomes, and then optimize the parameters. And so one problem with building like a cellular model is that there's a lot of cells, right, in the body. So for a particular disease, like cancer, cells are heterogeneous. There's a lot of different cell types involved, right? The disease tissue is very heterogeneous. So a lot of genomic profiling has moved on to single cell genomics, where you identify different clusters, different cell types in the data, and then from the expression of those single cell types, you could build networks, gene regulatory networks that could describe the change of one cell type to another, right? So, for instance, like from a cancer stem cell to a more mature malignant cell, right? And so you're kind of you can like build individual agents for each cell and then collect them into a multi-agent model. Okay, and then all right. So just to summarize, kind of the main challenges in terms of identifying targets. One is kind of the high number of cell types. Okay, so while you can do there's single cell sequencing technologies, it's still costly for profiling each cell type, and there are billions of cells, in a disease tissue. So you may never be able to capture the full heterogeneity of the disease, right? And there's also still, like I alluded to, with only MR being available, there's a question about like whether the targets are causal or associated with the disease, and then a lot of these studies are like comparing kind of healthy and very sick patients, right? Just because it's easier to get the samples from patients who are like very sick in the hospital, but we're kind of comparing kind of very healthy to very end stage, and we're not capturing the whole progression of the disease, right? So, like I mentioned at the beginning, that with chronic diseases, they occur over a long period of time. So for some cardiovascular disease or like dementia, it could be, decades, right, before we see the end stage of the disease, right? And actually for Alzheimer's disease in particular, we can't really actually diagnose them until they've passed away, right? You can actually correctly diagnose Alzheimer's disease until you actually see the brain's pathology. So you're really only looking at the end stage of the disease, and you're having difficulty getting samples from kind of maybe middle, intermediate stage, right? And with that, like a lot of samples, when we do the profiling, we're basically taking them from the patient, right? So they're already dying, right? So a lot of the information that we get from the samples, you have to realize there's what's known as a post-mortem effect, right? So with gene expression, it's very reactive. So even a few minutes to an hour, the gene expression starts changing when you take the samples out. And then also they're very reactive, especially gene expression, methylation, they're very reactive to environmental changes as well, right? For instance, smoking is a big one, right? I don't know if you have looked at clinical data, there's no studies where you actually have very clear smoking information about that individual, right, or that patient, the patients, right? And then that's a huge confounder. Okay, so after identifying the target, so if we have a reliable target, we want to identify the drug or the therapeutic, right? This is I'll like disclose, this is not my area. I don't really work on like drug design, but this actually slides from a pharmacologist that used to teach on a master's course that I led, but what you want to really there's several factors, right, that we want to optimize for drugs. I think a lot of the development in computational methods have mainly been focused on potency against the target, okay? But just to make you aware that there's actually other factors like toxicity of the drug, availability of the drug, absorption, like how your body deals with the drug, right? And selectivity, if there's like kind of off-target effects of the drug, and how easy is it to produce, okay, manufacture. And each of these other elements may actually determine the success, whether that drug makes it out to the market or not, okay? So it'd be awesome if you're basically, that's your objective, right? Like to maximize on potency, bioavailability, reduce on toxicity, reduce on chemicals, manufacturing processes. Okay, but in reality, people kind of, like I said, they overtune on potency, and that usually leads to more toxicity. Okay, because I mean it's kind of intuitive in a way. You have a drug that really hits the target, it does really well, but likely it's going to be hitting it might be hitting that target in tissue that you don't actually want to treat, right? So that causes kind of toxic effects, right? And then this is more like the reality. Okay. So there's algorithms out there that optimize on these objectives, right? Right now, I don't think they optimize. There's no real algorithm that optimize on all of those objectives. Like I said, most like the AlphaFold algorithms optimize on the target, binding to the target, and it's doing very well now. Like I think this year's paper, they show it's outperforming organic chemists, right? So it does very well, and I think compared to Alpha 2, they've integrated some diffusion models to improve the performance there, right? And there's many other approaches that you could tackle this problem, but the main thing is that you have to focus on optimizing on all of these objectives. Okay. So other things to consider for kind of identifying drugs is that oftentimes the target is not just one protein, right? So a lot of the protein folding challenges, the Cas challenges for like drug binding and so forth. It's a protein, but oftentimes in a disease, that protein does not exist in an aqueous solution by itself, right? It's often actually in combination, complexes with other proteins. And those complexes could change actually over time, right? So what the drug might be hitting may be actually even covered by another protein. Okay. Other things to consider, like I mentioned, the targets proteins could change depending on the mutations that the individual might have, variants, or environmental exposures may also affect the target, right? And then the other thing I didn't discuss, I'll show some examples of that, is drugs will also interact with other drugs. So this is becoming more of a problem now because as individuals are aging, they're living with multiple conditions. They're likely taking multiple drugs at the same time, right? So you may have a cancer patient that's taking anti-diabetic drugs, right? And those drugs, in the trials, a lot of times we don't model that, right? We don't even test that, right? So that's something. That's another layer of complexity that you have to consider. Okay, so after kind of identifying a decent drug candidate, you don't want to go straight to patients. Some other therapies you can go kind of straight to patients, right? Like maybe like surgical therapies. Surgical therapies they might test on animals as well, but like digital therapeutics and so forth, some you can go straight to patients. But for drugs, you generally want to do preclinical testing, and in terms of like one of the objectives, like the efficacy of the drug, you want to try to optimize this dose response curve, right? The idea is basically you want to give the patient as little drug as possible, right, but have kind of maximum effect. Okay, so this is like dose response curve for a cancer cells. So you're basically trying to kill as much of the cells with as little drugs. And the kind of measure of this response, one measure is the IC50, the inhibition concentration at 50%, the 50% inhibition concentration. And so when you're looking at this kind of data, you may get curves like this where you're increasing the drug, but there's no change in the number of cells, right? The number of disease cells. You might get a delayed response, or then you might get a sensitive response, right? And the objective here is like, can we predict this difference, right? Is there like features like say genetics that can predict this difference? Okay. So generally, with the preclinical testing, they'll basically test the drugs on, say, cell lines or animal models or organ models, and they will also profile those models, right, to identify what features of those models would predict a good IC50 value. Okay. So there's some publicly available data sets. This one is out of Sanger called GDSC. That has a panel of like 1,000 different cancer cell lines and kind of 400 different drugs that they've kind of systematically tested, and then they have also like genomic features on those cell lines. Right. And so Juan Jose in my team and co-supervised by Mauricio as well have developed a multi-output model to basically map the relationship of the genomics and chemical features of the drug to the individual doses of, sorry, oh, it didn't show up here. Okay, I don't know why there's supposed to be dose response curves, but basically we're trying to map these genomic features, high-dimensional genomic features to the dose response curves, okay, of each drug against each cell. Okay. And then so you can systematically test this across different cancer cell lines, right? Cancer types. We're doing that because we're interested in potentially therapies that can treat multiple conditions. Okay, not just so specific to one. And why it's a multi-output model is that we're trying to predict for each experiment. There's in here, there's like 7 outputs, right? One for each concentration. So one response percentage viability for each concentration. So we want to predict this all in one go, right? And generally, like previous metrics have just trained on single measures, summary measures like IC50 values, right? But if you don't look at the whole dose response, you could get cases where you have the same IC50 value and even the same area under the curve for this dose response, but they may have actually different responses, right? So other metrics like Emax may be different, right? So basically, it's I think key take-home point is that like with evaluating drug response, there's a lot of different metrics out there, and you may need to use more like multi-output models to try to predict the different aspects of response. Okay. And then there's a kind of a scaling problem, right? Because, like I said, not many people just take one drug at a time. Some people take multiple drugs. And there's actually even combination drugs could be actually helpful. Right, in cancer, you may want to, because say the tumor is heterogeneous, some cells may respond to one drug, some other cells may respond to other drugs. So you may actually want to combine the drugs. So then this problem gets harder because you're going to do that prediction on higher order manifolds here, right? So this is just 2 drugs, right? And then you have, instead of a dose response curve, you kind of get like a 3-dimensional volume, right? So to tackle these problems, you can use crowdsourcing. So this is a study a few years back with AstraZeneca where we basically released some drug combination data and asked the community to come up with algorithms to predict combination responses. And I'll just jump to the results here. Basically, people submitted different types of algorithms for predicting combination drug response. But what we saw is that there's certain combinations of drugs that all teams predict pretty well. Some drugs that like certain teams and algorithms did well on. But there's also like a proportion of drugs that no algorithm actually, no approach did well on. Okay. And so this is kind of showing more on like how we evaluated the predictions. So one metric is just to look at the predictions versus observed, the Pearson correlation between the predicted response and observed response. Right, and how we benchmarked that was using kind of like a random model, and also not a model, but actually replicate experiments. So the idea is that there's some variability in those assays, those drug tests, right? So the algorithms shouldn't predict better than you basically just repeating that experiment. Okay, and so what we saw is like actually there's quite a few. Algorithms that actually performed on the level of replicates. So that's pretty much as best as they can do. But the overall correlation, even between replicates, is quite low. Okay. So then you can look at the dose responses themselves, and then what you see even across replicates is that there's a lot of variability. Okay. And then, this kind of earlier work where we modeled the variability using GP regression, right? And basically saw that there's a lot of the assays were extrapolating the curves beyond the concentrations that were tested. And there, if you're doing like kind of biomarker discovery or prediction, it would be very challenging, right? Because like, your confidence interval is huge here. Okay, so some of the challenges of preclinical testing highlighted basically reproducibility, right? There's a lot of variation between even replicate experiments of the same drug on the same samples, right? And then and the other thing is drug efficacy models don't account for ADME, which is kind of like another modeling problem. I remember when I was working at AstraZeneca, our office had people who were working on efficacy, right? And half the office was working on ADME. Oh no, I broke this. This is gonna.
I can see it.
I'll have to move here. Oh, it actually works with one battery. Oh, it only has one battery. Okay, great.
It only works with one battery.
Okay. Okay. So yeah. So ADME is like pharmacokinetics. It's basically we're looking at what the drug does to the patient, right? But there's also the reverse process of what the patient does to the drug, right? So the patient could actually like metabolize and absorb the drug, making it less effective, right? And they're kind of two separate modeling efforts, and I haven't seen any like too much work like connecting the two. Okay. Any questions? All right. I think you are ready for the wine and posters. Okay. All right. I'll jump into patients. I think like a lot of people work with patient data. So in clinical trials, the main problem is like you want to select the patients, right? It's like the inclusion criteria for the trial, right? You don't want patients that are not likely to respond to the drug because it costs so much to run a clinical trial. So you want patients that could benefit. And so that basically looks at, you're looking at trying to identify the heterogeneity of the disease. You want to try to stratify a disease. So one disease I study is pulmonary hypertension. And it's really hard to stratify this disease and define like inclusion criteria and so forth, because it's like a disease that's, like many complex diseases are based off of more symptoms. So it's kind of a combination. It looks like a lung disease and a heart disease, but it's kind of defined by the symptoms, right? And it's kind of arbitrary. It has like arbitrary thresholds for like kind of arterial pressure of the heart, within the heart, to decide whether it's one form of pulmonary hypertension or not. So they're using these kind of symptoms to kind of decide who's going to be in a drug trial or not. So we're just basically looking at RNA then and proteins and seeing if there's any clusters. Okay. So this is like pretty just simple cluster analysis. Okay. So it kind of looks like there's some differences, heterogeneity, right? So even within one form of pulmonary hypertension called pulmonary arterial hypertension, you get some differences between individuals, right, in their outcomes, survival outcomes, PCA of their blood proteome. You may also get some clusters, okay, of individuals. So we evaluated kind of the blood biomarkers a bit further. So looking at the transcriptome of whole blood from these pulmonary hypertension patients, we developed a tool. Socrates, a PhD student, developed a toolkit called OMADA that it's not a new clustering method. It's just a toolkit that tries a lot of different clustering methods and outputs a lot of different clustering metrics, right? Like stability, silhouette score, compactness. There's a lot of different measures that you can measure how good the clusters is, right? And then it's kind of a little bit art form, right? You kind of like squint your eyes. You get a clustering output that's satisfies most of the metrics, and then you kind of plot the data and you squint your eyes and you say, oh yeah, there might be a cluster, and you try to publish it, right? And right now, like the validation is basically some patient outcomes, right? You're trying to see if there's actually any survival differences, even without treatment and also with treatment, right? So that's the state of the art right now. So it's not too far ahead. So there's a lot of room for development. The other aspect of kind of looking at drug effects in patients is drug-drug interactions. So I mentioned that people are on multiple drugs. So this is a database that we mined from. It's an HIV drug database in Liverpool, actually. I knew some people were from Liverpool, so they're well known for this drug-drug interaction database that was started in, I think, 1990s, actually. So they kind of curated, they just kind of mined all the clinical records and built up this database, well-curated database of reported drug-drug interactions, basically side effects from different combinations of drugs, right? And one PhD student just developed a simple CNN to predict the different outcomes of drug-drug interactions. Okay. I think the challenge is that the lines between like these different categories of drug-drug interactions are pretty vague and arbitrary. So it's like they get clinicians to kind of evaluate evidence of drug-drug interactions and give it these codes, like red light stop, right? Green light go. And then there's like 2 different amber lights, right? Or yellow and amber. And I think it was like very difficult to predict between like the amber and the red and so forth, right? So that I think the work here may be like actually looking at the data collected and maybe trying to create more automated approaches to classify or label that data better. A follow-up study that PhD students working on is kind of along the lines of trying to combine mechanistic physiological models with machine learning models. So in that aspect of ADME I talked about, so that's something that's not so much of a focus, looking at absorption, what the drug does to the body. Most of those models are very mechanistic kind of equation-based models, right, that basically have a deterministic model of how the different organs deal with the drug, right? So the problem with like applying machine learning approaches for that is that there isn't much data, right, on patients to really like to train from. So the idea is like there is some well-established physiological models out there, and you could run those models to generate some synthetic data that could be used to augment some of the patient data that's been collected, to then train a machine learning model to predict the pharmacokinetic effects. Okay. Just recently, some people have tried this, so this is like still more. It's pretty intuitive idea, but yeah, there hasn't been any like success stories so far. I'm not like a pharmacokinetic modeler myself, so I mean, there isn't a large number of physiological models out there. Okay, so it might be there's some assumptions that may need some work in these physiological models, and also these models are not super large as well. They do kind of abstract a lot of things. Okay. So now we're going a bit more into the data science type of things and looking at like monitoring. So after you get the drug into the market, right, you often need to still keep monitoring the patients to look at effect of that therapeutic, right? So one big challenge is clinical data. So like some of our cohorts, like pulmonary hypertension, we collect follow-up visits, data from follow-up visits, and this is basically the data. Like there's a lot of black, right? There's a lot of missing data, and I think there's imputation methods, like using more statistical approaches, like multiple imputation methods. But I think there's scope for some ML deep learning methods to try to impute that clinical data. But that's a very hard thing because each clinical variable is from a different source. They have different distributions and so forth. But there's opportunity here. So wearables is becoming a prominent source of clinical data. So this is actually a study where we looked at healthcare workers right at the start of the pandemic. actually mostly in Sheffield. So like from like April 2020, so like at the COVID outbreak. And we collected their symptoms over time. So healthcare workers, because they're kind of frontline people, they often got COVID, and then we tracked their symptoms, right? But we also got a good deal from Apple, got some free Apple Watches that we gave to them, okay? And I'll tell you right now, like a lot of them didn't appreciate it or something because there's a lot of missing data out there, right? So I think we gave out 200 watches. I feel like 200 enrolled in this study got watches. I think we got data points. I mean, the symptoms were pretty well collected. There's about like 200 individuals with symptoms. Or no, not 200. It's like we overlap with some of the activity data. So maybe 120 healthcare workers that had usable kind of longitudinal symptoms data, but only like around 40 healthcare workers actually had usable kind of activity data from the wearables. Okay. But the idea, like what we were trying to do was trying to see if we can like use physical activity to predict symptoms, especially long-term symptoms, i.e. like long COVID. Okay. The data's too sparse to do that. But we found some, interesting patterns, right? There does seem to be like 2 patterns of physical activity in the healthcare workers, and then there is 2 patterns of kind of long and short-term COVID symptoms, and they do agree with other studies that looked at COVID symptoms, like those specific symptoms like loss of taste. Like the prevalence of that seemed to agree with other epidemiological studies. So just looking at some of the challenges of looking at wearables data, right, is basically, like I said, even those people who actually use the watches we gave them, there's still a lot of missing data, right? So there are even times where they didn't use the watch or phone at all, right? This may be maybe sleeping time, or they may ran out of batteries, right? A lot of times they did not use the watch. We had the phone data. We later found out, actually healthcare workers, like if they're going to like the operating room, they cannot wear things on their wrist. So I think we didn't think about that when we gave them. Maybe we shouldn't have given them to like nurses that were working in like ICUs and so forth. So there's quite a lot of missing data. Can we impute that data? So we try to. So we try to deal. Oh, I'm missing a slide. It's a shame I missed a slide. I don't know why it got cut off. There was a slide showing basically we use multiple ways. You can scale the phone data to look like the watch data, or you could use a predictive model to train on the phone data and predict the watch data. So that's kind of the approach we use to kind of fill in these holes. Okay? But there is quite a lot of holes. So especially with wearable watches. So I think for these kind of studies, having multiple devices really helps with kind of this data processing and augmentation. Okay. Other types of longitudinal data is so we can omics, and also what I talked about earlier, like the body mass index, like routine physiological measures that we can get, right? So actually children, especially when they're born, because the NHS checks on them, there's actually plenty of longitudinal data on those children. So we're able to kind of get also some biosamples from children, right? And then look at, they're mainly from like cheek swabs of children. Actually, at an early age, you're not actually allowed to get blood. There's some more tighter ethics for getting blood. But you can get some biosamples from the children and then look at how methylation changes, right? Because like I said, one of the biggest confounders is these like environmental exposures like smoke smoking, and so forth, they really affect molecular profiles like DNA methylation and transcriptomics. So we want to look at that, but like if we're looking at older people, there's just so many hidden confounders, right? You don't know what they're doing, and you're not sure even what they did in their 20s, right? They might be taking weird substances. So we're really focusing right at the beginning of birth to try to see what environmental exposures could influence their methylation, right? And so so what we're using is a multitask GP to basically define mean processes for different clusters of methylation and body mass index trajectories, and then use those mean processes to do the prediction. Okay? I know Mauricio doesn't agree with doing multitasks, but there is some intuition around this that like there has been some epidemiological studies that have found that there are different kind of clusters or different patterns of growth amongst children, right? So it's not like so it's like there's some kids who maybe growing faster at the beginning, some growing slower, and so there is some heterogeneity even amongst children in how they grow and develop.
What were the yellow dots?
Oh, sorry. So the black dots are training. The yellow dots are the forecasted points. Right. So the utility of this is so you want to try to predict what their future growth trajectory may be like, because one like when kids go to adolescence, there's puberty. It's like there's a lot of physiological changes that happen, and then you want to see if some people develop faster, some develop slower. That also may factor into future cardiometabolic risk factors. And then we also try to do this with molecular data, like DNA methylation, try to forecast as well. One of the reasons because not like some clinical variables like BMI you can collect pretty routinely, but like biosamples, it's quite costly to collect, right? You may not be able to get your patient in to collect those samples. So if you had some historic methylation data, you could train models to then forecast future data. Then you can compute molecular phenotypes like epigenetic age. Okay, good. So I'll just finish up on what are some of the wider challenges for this type of work. So one is, I guess, not surprising. I don't know if everyone agrees with this. There's a survey, actually, I think 10 years back by Forbes, data scientists. I think maybe it hasn't changed, maybe it's gotten worse, but like data cleaning, curation, obviously takes up most of the time, right? Coming to this conference, this workshop is basically 4% of my time, right? Where we're actually trying to learn new approaches. Right, and then so that 60% is also not much fun as well, right? So there's some promise maybe using LLM models to at least for like the clinical data curation. The team at Google have shown that you can train LLM models to do better like labeling of clinical notes, unstructured clinical notes, right? So that should save some time for kind of curating clinical data. The other challenge is kind of arose through actually COVID, because I did some work on COVID outcomes, and you basically notice that actually a lot of the health outcomes are dictated by not the science, but more the behavior and social factors. So you could have very good treatments, vaccines, excellent vaccines, but what really determined the rates of infections outcomes was also a lot of behavioral and social factors, such as mask wearing, social distancing, and so forth. And so that's kind of not really integrated into this model. So like in Singapore, we basically reacted by hiring a lot of sociologists into our institute to try to develop more, use computational sociology to develop more models that can predict patient behavior. And next is like, how do we integrate that with kind of the molecular and wearables data that we collected? Okay. And then finally, I think the previous speaker, Alex, alluded to kind of multi-scale models, right? So we're building models for kind of cells, tissues, right? Some organs, right? Eventually, we'll have enough data maybe to build a whole patient, maybe. Maybe it's not needed. I'm not sure. But this isn't. I mean, this in the lab there. The preclinical testing is kind of doing that, right? Using animal models, you're making an assumption that animal model is representative of that patient. So the idea is like to develop a digital twin of that patient, of each individual patient, so that you can test hypotheses, test potential therapies on them. And that should hopefully personalize the care even further. Okay. All right. I'd like to thank team and collaborators and funding sources. All right. Thanks.